All right, let's jump right into this explainer. Today, we're unpacking a really fascinating, highly targeted laboratory protocol. We're looking at the exact mechanics behind what's basically an extreme survival test, one that's designed to isolate bacteria that can actually eat diesel oil. It's a rigorous, totally unforgiving scientific process, and we're going to walk through exactly how it goes down. So, think about the core challenge here. How do you actually find a microbe that metabolizes diesel? I mean, it's not like you can just look through a microscope and ask them what they like for dinner, right? We have to force these tiny microbial powerhouses to reveal themselves. And we do that by setting up a highly controlled, high-stakes situation in the lab. Which brings us to part one, seeking oil-eating microbes. What we are actively hunting for are called hydrocarbon-degrading bacteria. Basically, these are specific bacterial isolates that can prove they have the chops to grow on a medium where the only real food source is diesel oil. If they manage to thrive in that environment, they are officially flagged as potential degraders. It proves they have the raw biological hardware to break down those complex, stubborn hydrocarbons. Okay, let's move to part two, building the survival arena. Now, check out the exactness of this recipe. This is what we call the mineral salts medium, or MSM. You whip this up in an Erlenmeyer flask, and per liter of water, you're looking at 1.8 grams of K2HPO4, 4.0 grams of NH4Cl, and some very precise, tiny trace amounts of magnesium sulfate, sodium chloride, and iron sulfate. But here's the absolute kicker. Notice what's missing. There are absolutely zero standard carbon food sources in here. None. It's literally just base chemical compounds. To actually construct this testing ground, scientists follow three really strict steps. First up, you dissolve all those chemicals into 1 liter of distilled water. Then, step 2, you've got to perfectly balance the pH to a neutral 7.0. And finally, the whole mixture gets sterilized at a blistering 121 degrees Celsius for about 15 to 20 minutes. This guarantees you're starting with a perfectly sterile, complete blank slate before the real test even begins. Moving right along to part 3, the 7-day trial. For this primary screening, we use what's called the spread plate method. This is where each pure, isolated bacterium that we're testing gets physically spread directly onto the surface of that mineral salts medium agar we just made. We are basically dropping them straight into this carefully constructed, barren wasteland. And this right here perfectly highlights just how extreme this test really is. That agar they're sitting on, it's supplemented with a tiny, mere 1% diesel oil. Literally, just 1%. That is the one and only addition made to those base mineral salts. The reason for this comes directly from the protocol itself. The whole point is to screen for bacteria that can, quote, utilize diesel oil as the sole carbon and energy source. Because there is zero other carbon in that petri dish. This rule is absolute. If these bacteria want carbon and energy to survive, it's do or die. They either figure out how to metabolize that diesel, or they starve. Once they're locked in with that single tough food source, the plates go into an incubator, and the temperature is held constant at exactly 37 degrees Celsius. This nice, warm environment acts as a catalyst. It speeds up the biological activity, forcing the bacteria's hand to either successfully process the fuel right then and there or fail completely. And we aren't just leaving them in there for an afternoon either. These bacteria are trapped in this harsh, warm, 1% diesel-only environment for 7 long days. It's a full, week-long test of extreme dietary deprivation to see exactly who has the biological mechanics to tough it out. Which brings us to the final stage, part 4, identifying the winners. At the end of the week, the results are beautifully definitive. You see, the mineral salts medium acts like a flawless, uncompromising filter. Because the environment was so perfectly controlled, only the bacteria that were actually able to grow on that diesel oil-supplied MSM survived the week. Any isolate that managed to multiply and form visible colonies, well, they've unequivocally passed the test. By simply staying alive, they've proven their unique capabilities, and they officially earn the title of potential diesel oil degraders. And that wraps up this incredibly focused laboratory protocol. We've just seen how scientists literally strip away every single luxury, leaving nothing but mineral salts and a tiny drop of diesel, just to force nature's microscopic cleanup crew into the spotlight. But you know, this leaves us with a pretty fascinating final thought. Now that this rigorous screening has successfully isolated these potential degraders, what happens to these microbial victors next? I mean, how do we take them from a sterile petri dish in a lab and actually deploy them to clean up oil spills in the real world? It's like a giant puzzle. Keep that in mind as you think about the incredible hidden power of the microscopic world. Thanks for tuning into this explainer.